Hello friend, myself Professor B.D. Bhoknal, Associate Professor, Botany Department, KTCM College, Nasik. Now today's subject is Cryptogamic Botany, paper first within that now the Pteridophyta. Now today's topic is internal structure or internal characters of Pteridophytes. Now the internal characters of Pteridophyte, the now the xylotum, then now Selaginella and Marcellia. See, xylotum, it is from Pteridophyta. Xylotum, it belongs to class Xylopsida. Now, Xylotum stem TS. Now it is differentiated into epidermis, cortex, and central cylinder. Epidermis, one cell thick. It is not continuous, it is interrupted due to the presence of stomata. Epidermis, which is followed by many cell thick region known as now the cortex. Now cortex is differentiated into three regions: outer cortex, middle cortex and inner cortex. The outer cortex, it is made up of chlorine-chioma cell. It do the function of photosynthesis. Now the middle cortex, it is generally sclerenchymatous cells are generally thick walled. They have lignin deposition. Then innermost cortex, it is made up of parenchyma tissue. The cells are generally thin walled and they are compactly arranged. Then these many cell thick region, which is followed by one cell thick region known as endodermis, and one cell thick now pericycle. Then now the central cylinder stele is now protostelic, or here stele is siphonostelic because at the center pith is there. Then surrounding the pith, star-shaped xylem is there, and the star-shaped xylem, which is now overlapped by patches of phloem, and such type of stele is generally known as now Siphono in case of xylotum now stem. So xylotum now stem. TS epidermis is single layer with stomata and cuticle. Outer cortex is chlorine-chimatous. Middle cortex is sclerenchymatous. Inner cortex is parenchymatous. Endodermis is present. Pericycle is also present, and stele vary along the plant axis. Protostele or actinostele at the base, siphonostele in the middle of upper part. Sclerenchymatous pith is often seen in very old stem. Protostele, central solid core of xylem surrounded by phloem. Actinostele, star-shaped central core of xylem surrounded by now the phloem. Xylotum, now the TS of Synangia. Now see here, in case of xylotum plant, it has undergone structure known as rhizome. Rhizome, it gives rise to rhizoids only. True roots are totally absent in case of xylotum. Then aerial axis. Aerial axis, it is now generally shows scale-like structure known as now the lip. Now the axis or stem, it is generally dichotomously branched. Then the spore producing organ, it is generally present at the top of the plant body. Spore producing structure in case of the xylotum known as synangia, because three sporangia grouped together and this structure it is generally known as now the synangia. All spores are similar in size and shape. So the presence of homosporous condition now in case of synangia. Synangium is a trilocular or three-chamber spore-bearing structure in xylodon. It is considered as a fusion product of three sporangia. Location: axils of foliar leaves on aerial shoot. Synangium ts consists of three chambers or locule. Wall of synangium is 3 to 4 layered. Thick outer wall forms the epidermis, and the inner wall separates the three locules. Each locule is filled with large number of bean-shaped spores, and the spores are homosporous. Synangium splits along three longitudinal lines of dehiscence on the locule, thereby releasing now the spores. Then next, Selaginella now stem TS. Internal structure of Selaginella stem TS. Selaginella stem, see within this diagram, Selaginella stem TS. Now it is divided into three regions: epidermis, cortex, and central cylinder. Epidermis, one cell thick region. Cells are compactly arranged without intercellular spaces. This epidermis it is lined outside by means of thick cuticle. It checks the water loss from the plant. One cell thick epidermis which is followed by now many cell thick region known as now the cortex. Cortex is divisible into two parts: outer cortex and inner cortex. Outer cortex is sclerenchymatous. The cells are thick walled, have lignin deposition, and the cells are compactly arranged. While the inner cortex it is made of parenchyma tissue. The cells are loosely arranged, and the cells are generally thin walled. Many cell thick cortex which is followed by now a one cell thick region now known as the endodermis. The endodermis it gives rise to many hair-like structure known as now trabeculated endodermis. Then at the center, now the stele is protostelic. Xylem is present at the center, and the xylem which is surrounded from all sides by means of phloem patch, and such type of stele is generally known as now the protostele. Selaginella now stem tears. Anatomically, it is differentiated into an outer layer of epidermis, middle layer of cortex, and centrally located stele. Outermost one cell thick epidermis consists of cuticle-sized cells with no stomata. In most of species, the cortex is differentiated into a few outer layers: a thick walled sclerenchymatous hypodermis and many inner layers of now thin walled parenchymatous cells, while in very delicate species it is fully composed of thin walled parenchymatous cells. Cortex is completely sclerenchymatous in xerophytic now the species. Generally, there is no intercellular space in the cortex. Centrally located stele is connected with cortex with the help of many long radially elongated cells called as trabeculae. In between these trabeculae there are present many air cavities or big intercellular spaces. That these trabeculae are endodermal structures is shown by the presence of many band-like Casparian strips now in them. Trabeculae are absent in species like now these Andunuka species. Then the number of steels is variable from 1 to 16 as under. In case of Selaginella spinulosa, one stele is there, and such type of species is generally known as monostelic, while in Selaginella kraussiana, two steles are present within single stem, that is known as distelic type of species. Two steles are found within the single stem. Then in case of Selaginella, now Levigata, 16 steels are observed, and such type of species known as polystelic type of species in case of Selaginella. Next, Selaginella stropulus ls. Selaginella stropulus ls, it has central axis. Central axis, it bears two types of sporophyll, megasporophyll and microsporophyll. Megasporophyll consists of megasporangia, microsporophyll consists of now the microsporangia. Now the megasporangium is larger in size. Megaspores are few in number and the spores are larger in size, while microspores are smaller in size and they are many in number. Strobilus. All the species of Selaginella form the strobili or cone generally, terminally, side branches, but some species apical meristem of the cone by now continuate meristematic activity producing a foliage leaves. Selaginella is heterosporous and therefore sporangia are of two types, microsporangia and megasporangia. The sporophylls associated with these two types of sporangia are designated as a microsporophylls and megasporophyll respectively. There is variation in distribution of sporangia within the strobili of different species. The lower portion of Strobilus consists of megasporangia and the upper portion of microsporangia. In some species of one side of Strobilus bear microsporophyll while in another side megasporophylls. A sporangium, mature sporangia are stocked with a two-layered jacket. The microsporangia are slightly elongated and reddish to bright orange in color. Megasporangia are larger than microsporangia and are frequently lobed. Megasporangia are whitish yellow or light orange in color. Then the next and last, now the habit of Marsalia. Marsalia, now it belongs to now the class Pteropsida. Now see, in case of Marsalia, there is an underground structure known as rhizome, and the rhizome, now the nodal lower portion it gives rise to now the true roots. In the upper portion it gives rise to now the petiole. Then the spore producing organ, it is present in the axil of leaf. Leaf is pinnately compound. Leaf is generally now young leaf, it generally shows circinate vernation. Within young leaf, coiling is there, and such type of thing, it is generally known as circinate vernation. So this character, it is also present in case of now the gymnosperm, that is in Cycadophyta. So it generally show the close resemblance with Todus gymnosperm. Now TS of rhizome, internal structure of Marsalia rhizome. Now the epidermis, it is the outermost limiting layer of single cell thick parenchymatous cells. Stomata are absent. Then the cortex, it is differentiated into three regions: outer cortex, middle cortex, and inner cortex. Outer cortex, it is present just below the epidermis, also called as hypodermis. It is parenchymatous and may 1 to several cells thick. Some of the cells contain now the tannin. Middle cortex, it is also called parenchyma. It lies below the hypodermis. It consists of large spaces separated by one cell thick parenchymatous septa. Inner cortex, it is solid tissues of several cell thickness. The outer layer is thick walled parenchymatous, while the inner layer cells is thin walled, that is parenchymatous. Compactly arranged. Some of these cells are filled with starch or tannin. Then the stele. Stele is now siphonostele. Amphiploic siphonostele. Now in case of amphiploic siphonostele, now pith is present at the center, and the pith which is surrounded by now the phloem. Then phloem is surrounded by xylem, and xylem is again surrounded by phloem. So xylem is sandwiched within the now two patches of phloem, and such type of stele it is generally known as amphiploic siphonostele. So from the both sides of now xylem, two layers of phloems are present, and such type of stele it is generally known as amphiploic siphonostele. Now see within this diagram, the Marsilia, now this diagram, Marsilia, now rhizome, TS of rhizome. TS of rhizome, it is divisible into epidermis, cortex, and central cylinder. Epidermis, one cell thick, compactly arranged. Then many cell thick region. Now the cortex, it is divisible into three. Now outer cortex, middle cortex, and inner cortex. Outer cortex is parenchymatous, middle cortex is sclerenchymatous, and inner cortex is parenchymatous. Inner cortex, it generally shows now starch grain or tannin cells are there within now cortex. Many cell thick cortex which is followed by now the endodermis and pericycle. Now within the stele, pith is present at the center, which is parenchymatous. Then the pith, now which is surrounded by inner endodermis. Then inner phloem. Then the xylem. Then xylem is surrounded by now the outer now this phloem, and now the outer endodermis. So xylem is sandwiched within two layers of phloem, and such type of now the stele is known as amphiploic siphonostele. Amphi, it means on both sides of xylem two patches of phloem is there, and such type of stele is generally known as amphiploic siphonostele in case of now Marsilia. Next, now this Marsilia sporocarpellus, horizontal longitudinal section, cuts each sorus transversely, and it is seen that each sorus is an now elongated structure covered by delicate indusium. The sori are gradate, basipetal in arrangement, with rows of largest sporangia, that is megasporangia at the top, and 2 rows of smaller sporangia. It means the microsporangia on 2 sides. And such type of arrangements of sporangia known as basipetal type of arrangement of sporangia, and it is known as graded type of arrangement. The mucilage ring is present in the form of 2 masses, one in the dorsal and other in the now ventral side. Marsillia sporo. Vertical transverse section of sporocarp shows only 2 sori on 2 sides. Both the sori contain megasporangia. If the section is taken through the megasporangia, or the sori contain only the microsporangia. If it is taken through the microsporangia, the sporophore Sporophore is seen in the form of 2 masses on either side. The mucilage ring is present only on now the dorsal side. Now Marsillia sporocarp. Now see within this diagram, outermost epidermis, then on both sides now gelatinous ring is present towards the upper side as well as towards the lower side. Then now the sorus. Then the sorus consists of now the placenta at the top consists of megasporangium, and at the base now the 2 smaller microsporangia. Such type of arrangement of sori known as gradate type of arrangement, because large sporangia is placed at the top. Then the Marsillia, now the sporocarp. Marsillia, it is heterosporous. It produces 2 types of spore, microspores and megaspore. Microspores are smaller in size, megaspores are bigger in size. Then Marsillia sporocarp TS section goes through only the now megasporangium. So within sorus section goes. This section it only shows the single sporangia, because section it goes through only particular through that angle only megasporangia are generally seen. Then see within this diagram, Marsillia vertical section TS of sporocarp, it generally shows mucilage ring, then now the sporangia, sorus and indugium. Thank you, and that's all.